So I'm going to talk a little bit about some approaches that are being developed for image-guided therapy. My background is in a combination of particle formulations. When I was in graduate school, I worked with some of the preeminent minds on that, Patrick Staten and Alan Hoffman, who have been working on triggerable carriers as well as protein engineering for years. But my primary advisor actually was a physicist, or is a physicist, and he began to apply his knowledge in bubble dynamics and acoustics to biomedical applications. And so I've begun to sort of merge the two fields, and this idea of image-guided therapy has really begun to take off around the world with the advent of a lot of novel particle formulations. Molecular imaging has really taken off, and so it's become a really hot area in the last several years. Okay, so just to give it a backdrop, non-invasive imaging is a powerful tool that no longer is being used only for diagnostic applications. The ability to visualize the delivery of drugs, and we've heard of nanomedicine from the context of drug delivery systems earlier today, and you want to have some sense of how successful were you in actually delivering those drugs to the disease site, or the targeted site. The ability to do that has major implications, and just the way you schedule your delivery of drugs to the patient. The incorporation of diagnostic imaging methods into therapy will, I believe, lead to a paradigm shift in the practice of medicine. And while there have been, or there are many technical challenges that do remain, the foundation has been led for future success. So in this talk, I'll provide a flavor of novel approaches for image-guided therapy that are currently under development. And the nice thing is that imaging technology is quite a mature field when you think about it. There's MRI that's been around for years, for generations. Ultrasound has been around, I believe, for even a longer period of time than that. And so the ability to rethink imaging and apply it in a very new way is really been driving this whole idea of image-guided therapy. So I want to actually take a couple of stories or vignettes of how this is being used. And my laboratory, my group is predominantly focused on cancer and treatment of cancer, and in some ways also coming up with new formulations of ultrasound contrast agents to be able to detect angiogenesis that plays a critical role in sort of the evolution of a tumor. So I want to actually sort of bring this story back up. The Professor Zhang had mentioned earlier, but I'm not going to spend a whole lot of time on this, is the fact that there is, 1995, as he mentioned, was the approval, FDA, for liposomal doxorubicin. And so doxorubicin does have itself, if you inject it alone, does have a myriad of adverse side effects, including neutropenia, myelosuppression, which is basically suppression of the immune system, and then organ-specific toxicity, and more importantly, doxorubicin is very toxic to the heart if given in too large a dose. And so encapsulating doxorubicin in liposomes does protect the heart from its toxic effects. And so in the US, it's referred to as Doxil, this liposomal doxorubicin, and overseas it's referred to as Kalex. So the tumor can be passively targeted just due to the fact you have leaky vasculature at the site of the tumors, and so you can get passive accumulation of this payload at the tumor specifically. And so the dox release, based on the kinetics, you can have a positive tumor response. But the key word there is the kinetics. Okay? And so doxorubicin, there have been a number of publications that have suggested that there's a slow release rate on an average of less than 10% per day. And so because of that, the thought is that liposomal doxorubicin is not much more efficacious. You do protect organs from its toxic effects. You can protect other bodily features from its toxic effects. But the thought is that it's not more efficacious than free dox. If that was not the case, then people probably would not be working on other formulations for nanoparticles for drug delivery. If this was the end all to solve and resolve all issues associated with drug delivery and chemotherapeutic agents, we probably would not be looking at pH-sensitive liposomes and thermosensitive liposomes as formulations for localized and, more importantly, triggerable drug delivery. So my group has been working on the development of a thermosensitive liposome and actually trying to enhance or accelerate the release rate in a very controlled manner. And so thermosensitive liposomes have been around since, I would say, the late 70s. The idea is not exactly new, but the original formulations require a fairly high thermal dose. And a thermal dose alone can be harmful. You can end up with irreparable damage to healthy tissue. And so the onset of damage, this study was published some years ago. The onset of damage for the thermal dose based on this equation here, where 43 degrees is sort of your reference temperature. The onset of damage at 0.5 to 30 equivalent minutes at 43 degrees, moderate at an hour to 240 minutes, and severe at greater than 240 minutes. And so we have been working on a smart polymer that basically takes two different monomers. One is a temperature-sensitive or thermal-sensitive monomer, is isopropyl acrylamide, and NIPAM is typically what it's referred to as. And then taking a pH-sensitive monomer, propyl acrylic acid, and basically copolymerizing them. So we have a copolymer from this. Now this copolymer is pH-sensitive, and it is also temperature-sensitive from the same polymer. We're using a RAF technique in order to control what the polymer length is, as well as ultimately what the molecular weight is, and to try to produce a copolymer that has very low polydispersity. Okay, so you have a narrow size distribution, or I should say a molecular weight distribution. And this is in collaboration with Professor Staten and Dr. Covertine at University of Washington. And so we're post-inserting these monomers. We have a hydrophobic, it's terminated with a hydrophobic group. And so we're post-inserting these into liposomes that we have preloaded with doxorubicin after the application of heat for this particular talk, but you can also initiate a conformational change in the polymer by also lowering or increasing the acidity, so lowering the pH. You ultimately end up with this disruptive polymer that then disrupts the liposomal shell and ultimately leads to your liposomal contents. So the doxa release is a function of temperature. This is the NTSLs is a formulation that's comparable to Doxil. The yellow circles is for traditional formulation of thermosensitive liposomes. And you get significant release once you get above about 42 or 43 degrees Celsius. And recall, you start to see potential damage to healthy tissue if you increase the temperature beyond 43 degrees. And you hold it there for longer than half a minute. And these results are from exposures that were for at least 5 minutes. So you're holding the temperature above 43 degrees for more than 5 minutes in these cases. And this is all done with a water bath. So these are all in vitro studies. And then for the polymer modified thermosensitive liposomes, we're able to accelerate and also improve upon the release rates, essentially shifting this to a lower threshold temperature for release. And so instead of requiring at least 42 degrees Celsius to get roughly 40% of your drug out, we only need about 39 to 40 degrees Celsius in order to get that. Now recall, it not only has a temperature-sensitive component, but also a pH-sensitive component. And so by reducing or lowering the pH, we also can further shift this to a lower temperature. And so if you imagine, as has been measured by researchers, the pH inside of a tumor is actually lower than what it is in healthy tissue, by an order of about half a pH unit. And so if you can imagine or envision creating a polymer that will respond not only to the lower pH of a 6 or 6.5, as well as being able to heat that in a very controlled manner, you can actually get a lot more drug out in a shorter period of time inside the tumor. So that's the motivation for this particular project. Furthermore, we were able to significantly reduce by 2 orders of magnitude the thermal dose that was required in order to get at least 50% of your drug out. Okay? So we're looking at more than 5 equivalent minutes at 43 degrees. If you're using a traditional temperature-sensitive liposome, we're able to reduce that to 0.05 minutes, equivalent minutes at 43 degrees. Okay? So 2 orders of magnitude reduction in the thermal dose required in order to get drug release. Furthermore, we did some studies to compare or to assess what its performance will be in serum-containing media. And one of the challenges with drug delivery is actually making a particle, okay, that does not destabilize in the presence of serum. And that is a huge challenge. Okay? Not only destabilization, but as well as aggregation. So it's critically important to try to make a particle that maintains its structure as well as maintains its drug. upon injection. This is systemic injection. Okay, so that's one of the challenges. So in serum-containing media, our liposome was actually able to perform better than the traditional. So that's the white bar, okay, as well as, and we were able to also see a pH sensitivity by changing the pH from 6.5 to 5.0 compared to a pH of 7. So ultimately, what you would like to do is inject this into the body, and as Professor Zhang had mentioned earlier, have it accumulate in the tumor, okay, and then be able to change the temperature, elevate the temperature locally. Now the question is, how do you do that, okay? I want to show this. So the idea would be deliver this, accumulate in the tumor cells, apply some sort of energy to change the temperature, because dipping a person's body entirely in a water bath is probably not going to be the way to go. All right? So you want to try to do this more locally. And most of the animal studies that you will read is basically taking a mouse and dipping that mouse's hind limb or its entire body into a heated water bath. We're not going to do that with humans. So how do you actually deliver the energy in order to change the temperature locally? So there's a couple of approaches you can use. One is just radio frequency heating. And so you have an interstitial probe that you insert directly into the tumor. And then these antennas, you can basically submit or transmit energy through these antennas and be able to heat locally where the tumor is at. Now, the problem or challenge with that is it's minimally invasive. That's the first thing. Number two is depth of penetration. So is there potentially a limit on the types of tumors that you can actually heat locally? One case in point would be brain cancer. Okay, are you going to insert this directly into the brain? Is there another approach to actually heat the tumor locally? So what my group has been working on and working with a collaborator over at Harvard Medical School is with MR-guided focused ultrasound. And this technology has been around for a number of years. But the nice thing is, for one, ultrasound penetrates tissue fairly easily. It's fairly deep penetration, and so you can heat the liver, you can heat the kidneys, right? You can heat the pancreas fairly easily. Okay? And so all you really need is a water coupling station. And the patient, this is something that can be done as an outpatient procedure. So you do not have to have a whole surgical team in order to do this sort of process. And I've actually seen this type of energy delivery over in England, where they only needed about 2 people in order to run this whole system. So we first tested to make sure that we could use an attenuative medium, so suspending our liposomes in an attenuative medium that absorbs ultrasound, changing the temperature. We also wanted to have some sense of what the acoustic parameters were going to be or required in order to get triggered release. And we were able to heat in a very controlled manner and sustain the temperature. That's the other key point, is not only being able to heat, but also sustain without going over a prescribed or predetermined temperature point. So if you want to achieve 43 degrees, you want to hit 43 degrees and then maintain that temperature of 43 degrees for longer than 5 or 10 seconds. You want to be able to heat to that and sustain and maintain that temperature for on the order of a couple of minutes. So we're able to do that, and we were able to also, once again, get trigger release. The TSLs is the specially formulated ones versus NTSLs, which is a melting point of about 57 degrees Celsius. And so we did not get release just from the ultrasound exposures alone. So with MR-guided focused ultrasound, the nice thing is you can measure temperature changes. So what's referred to as a proton resonance frequency. Proton resonance frequency is temperature dependent. So as long as you know what the reference temperature is, what the starting temperature is, you get a reference image. As the proton resonance frequency changes and you have a 2-dimensional image of that, then you can quantify what the change in temperature or temperature elevation is within that plane of view. And my colleague that works with this technology tells me he can get about 1/10 of a degree Celsius resolution or precision in terms of measuring that temperature change. Okay? So we worked on trying to determine what the acoustic parameters were going to be. Usually with these focused ultrasound devices, you end up having your energy is focused down to about 1 millimeter in diameter. Okay? And so my colleagues have a specially designed and custom assembled transducer that allows them to heat about 1 centimeter in diameter of tissue. So they're able to heat a larger area and a larger volume in a shorter period of time. The other nice thing was by using MR thermometry and using that as some feedback guidance, they were able to come up with the acoustic parameters in order to maintain a temperature of 40 degrees Celsius, starting temperature of 36 degrees, so heat by 4 degrees, or heat up to about 43 degrees and maintain for about 5 minutes. So a 3-degree difference by just simply changing the acoustic parameters, monitoring the temperature change. This arrow is pretty much the focal plane for the transducer. Okay. But changing those acoustic parameters, being able to change the temperature elevation and monitor that and get a 2-dimensional map of what that temperature change is. It's actually not the one I wanted to show. So we're actually currently finishing up studies on that in vivo, and we actually just finished our data collection. We're trying to now quantify how much drug was released locally. We know what the temperature change is and what the benefit or the advantage was by actually being able to locally heat the tumor and get drug release and look and see if there's a difference in the tumor growth or tumor regression. So we're actually currently trying to quantify that data. I was hoping to have that data to present to you today. The one thing I have been told is for our polymer modified liposomes compared to conventional thermosensitive liposomes, heated up to 43 degrees and roughly the same amount of drug, the tumor grows at a much slower rate. And actually with our polymer, it grows at roughly, tumor growth is repressed, comparable to if you were to give a free injection of free drug. So we actually believe we're able to get more drug out inside the tumor using our polymers compared to conventional thermosensitive liposomes. Now, if you can, I've had questions have been posed to me, if you can heat the tumor with ultrasound, why don't you just heat it to a point where you cause irreparable damage? And so that's something that my group has been working on as well. And so you can actually denature proteins, the process referred to as thermal necrosis, coagulative necrosis. You can heat the tissue to a point where you form a lesion. You denature the proteins, it gets much stiffer, you kill the cells. And so this is just a video showing, for one, the precision that you can get. This is, I believe this is an eyeball from a cow. And so the nice thing is it's optically transparent, but it also contains proteins. And so we can heat that preferentially in the center and form a lesion. Okay, and this is roughly the same shape and dimensions of the hot spot or the hot zone. Excuse me, the point where you have the focus energy is at its greatest inside the beam path of the ultrasound transducer. So the idea then is to form these lesions, and this is propagating the energy through intervening tissue. Form the lesions specifically inside the tumor. So this is an example of what you would like to do in liver cancer. And by moving the transducer focus around, you can change the location of the lesion formation. And so you can ultimately debulk a large volume of tumor by just changing the location of your focus for your transducer. So this has been used clinically to treat patients with breast. They've gotten much further than this over in Asia as well as in Europe. So they've treated with great success breast, prostate, kidney, liver, and sort of the new direction is pancreatic cancer. Because as most people know, pancreatic cancer, if you are diagnosed with that, that's almost a death sentence. I think Steve Jobs has still been dealing with that for roughly 6 or 7 years. I think he just recently had to step down to take a leave of absence from his job with Apple just because he's still dealing with this very deadly disease. Okay, so however, the cooling period for doing this, you can imagine if you're heating the tumor, you have perfusion, you have conduction. You not only, you are preferentially heating a small spot, but heat will dissipate. Heat will migrate and move around to other areas, so the surrounding tissue will also heat up. Okay, so most of the approaches have been based on heating for a predetermined length of time, predetermined amount of energy, assuming that you're starting with the same initial temperature. Okay, so the assumption is I'm always going to start at 37 degrees. The more energy you deposit, the higher the temperature is going to be, starting temperature is going to be inside the tumor. So you think you're heating, your initial temperature is 37 degrees, and it's probably more like 45 to 50 degrees Celsius. Okay, so there's an issue that in order to allow the tissue to cool back down, you have to wait about 2 minutes in between each exposure. And so ultimately your treatment takes a few hours in order to complete for an entire tumor. So with MRI, you can actually get some guidance of where exactly you want to heat because this is a non-invasive procedure. So unlike surgery, you're not actually opening up the patient. This is directly through the skin into the target tissue. And so you can measure what that temperature change is, and by looking at a change in contrast within an MR image, you can get some sense of where the ablation has occurred, and this is just a corresponding excise of the tissue of the organ to show that these dimensions are roughly the same. So MR, once again, image guidance provides you that capability to do that. So MR thermometry does provide 2D maps of temperature elevation, but it does suffer from major shortcomings. First and foremost, images are typically acquired first and then processed, so post-process. So it's not exactly real time. Okay, so that's the first thing. Second thing is motion due to breathing, depending upon which organs that you're trying to target, say the liver or the spleen, you'll have some motion artifact. So that's another problem that people are trying to deal with. And with animal experiments, what they oftentimes will do is just regulate breathing for the animal. So they actually will try to control lung expansion for the animal, so they can synchronize when they actually send the pulses. Finally, this 2D measurement, you're trying to conduct this 2D measurement and get information for a 3D phenomenon. So you're imaging within a plane, you're making your measurement for that plane, but you're actually heating in 3 dimensions. And it's typically an ellipsoid or a cylinder that you're heating. So you're trying to infer 3-dimensional phenomena from 2-dimensional information. Okay? So while temperature measurement is important, the detection of the ablation is actually more valuable. So you can actually detect the ablated volume in quasi-real time. That will be more informative for the clinician. And you can use that for feedback control. So the question is, how do you do that? So one of the professors in my department has been working on this idea called Acousto-Optic Imaging. Okay? And so what he would like to do is determine when you have this lesion that forms, where exactly is the lesion inside the organ relative to your beam focal point, and then how large is the lesion. So monitoring does require imaging. MR thermometry, as I've stated, is the gold standard currently. And the other approach that people have used, particularly over in England, is to watch for boiling. So you can heat tissue, you can heat an organ beyond the boiling point fairly easily. You can actually do it in less than a second. Okay, depending upon if you have enough energy you shoot into the body, you can actually get to 100 degrees Celsius on the order of milliseconds. You don't want to necessarily boil every part of the body, so you want to be able to control that. So preferably, you like to work with lower power levels. So currently what they do is look for these echogenic spots on ultrasound images, and that is an indication that you've achieved boiling. So you're basically over-treating the tissue. If you do not have bubble formation, then the question is, where exactly is your lesion in an ultrasound image? There's not enough contrast. Okay? In ultrasound, one of the benefits is it is basically real time. So the idea that my colleagues have been working on is this acousto-optic, where you actually, when you're shooting in your ultrasound pulse, you simultaneously send in laser light of a particular wavelength. That bounces around, of course, inside. You get a lot of diffuse scattering that goes on inside the tissue. But if you're able to change the index of refraction in a very controlled manner, and that's all ultrasound does, it changes the density locally. So if you're changing the density, you're changing your index of refraction. So you're basically phase modulating the photons. Then you detect these phase modulated photons on the other side of the tissue. And you create an image from that based on comparing your phase modulated photons to a reference image. Okay? So you actually get very good sensitivity to be able to create a lesion. Create an image. So he's been working primarily with with phantom material. And so this is just an example of the setup. And he initially was working with a B-mode ultrasound imaging device. So fairly low pressures that were being introduced. So this is the type of, this is a 2-cycle pulse that you can get out. This is the image or the, this is the projection of what the photodetector would see. And if you put an occlusion in there, then you get additional scatter or loss of light at that point of the occlusion, and so you can actually pick that up on your image. Now, if you create a lesion, you're actually changing the optical and mechanical properties of the tissue. You're actually making it stiffer, for one, so you're changing the stiffness of the material. And number two, you're actually increasing the scattering inside of the lesion, so you're actually losing more light. So this acousto-optic technique, they've been able to create these images, and so they wanted to come up with a method where they can monitor the on point, so turning on the HIFU transducer, turning off the HIFU transducer creates, turning on the HIFU transducer actually reduces the amount of light that actually gets through the phantom material. And so by modulating this and taking the RMS value for that, connecting it up to a lock-in amplifier, they're actually able to take one data point and monitor the voltage or the change in voltage over time. So this is actually going to be real-time imaging. So this is ultrasound, diagnostic ultrasound, trying to detect a lesion that's formed in tissue mimicking phantom material. As long as there's no boiling, there's not enough change in the density of the material to provide enough contrast in the ultrasound image, so it doesn't show up. And actually, I believe this was excised chicken breast that they conducted this test on. But with the acousto-optic technique, they were able to change in the intensity, so essentially the amount of light that actually gets through to the photodetector, and then monitor that over time. And in this case, they were able to detect a lesion that was formed using the acousto-optic technique where they were not able to detect it using ultrasound, standard conventional B-mode ultrasound. So these were my colleague likes to call this a tale of three lesions. The first one is actually no lesion is formed, so there's no change in the acousto-optic signal. The second is there's a change in about 55% reduction in the acousto-optic signal. And the third is a change of about 70%. And they tested to see what the correlation of that was for the volume or the size of a lesion that was formed. They did this using a number of parameters and got a fairly good fit, provided the lesion that was formed was below a particular size. So you get really good correlation. Now the question is, you have good correlation, and the other thing I need to point out is they use different parameters. So I had previously mentioned for the current standard practice for implementing this approach for HIFU ablation is to use a predetermined power level and to treat for a predetermined length of time. Now the question is, do you always get the same size lesion? Because all of you who work with animals or work with human specimen know there's biological variability. So due to biological variability, the answer to that question is no. You can use the same exposure conditions and get a different size lesion. Okay? So what you would like to have then is some feedback control to determine when you need to turn off your power source, okay, or when you need to change the exposure levels of your power source in order to reproduce roughly the same size lesion. So they're using this technique in order to do that, and they built this feedback control system. And actually we're able to, this is a fixed duration, fixed pressure level, 10 megapascal. And this is the variability in the size of the lesion as a function of exposure durations. With AO guided, so acousto-optic guided ablation, they're able to reduce the variability in the size of the lesion up to a particular size. Okay, so they're taking the information and using that to control the exposure conditions of the HIFU transducer. So there's a 30 to 40% decrease in standard deviation in the lesion volume with AO guidance, which ultimately leads to more consistent lesions that are formed. Okay, and then excised, this is all done in excised relatively homogeneous chicken breast. So a current idea is to move this into animal studies and test and see how effective this is in real tissue. So in summary, the acousto-optics is a technique for sensing optical properties in diffuse media. And the key word here being diffuse media. So you only need to actually capture a few photons, which is an advantage of this technique via the interaction of light and sound. The spatial resolution of ultrasound you actually can achieve using this approach, and the spatial resolution of ultrasound is on the order of about 100 microns or so, depending upon the frequency. Okay, so you're getting that spatial resolution. If you turn up, you increase the frequency, you can actually reduce the spatial or improve the spatial resolution. The candidate organs for this technique are breast, brain, prostate, and skin. You can do dual mode imaging using B mode ultrasound to pump the AO response instead of just the power transducer that you're using for ablation. And most importantly, you can get real-time acousto-optic sensing of HIFU lesions. So you're not detecting the temperature change. You're detecting what you actually ultimately want when you're using HIFU, or radio frequency for that matter, or cryoablation. You're actually detecting the lesion formation in real time. Okay, I think the last thing I wanted to talk about, how much time do I have actually?
I'd say about 4 more minutes.
4 more minutes? Okay, the last thing I want to mention is, in addition to the liposomes my group is making, we're also making perfluorocarbon nanoemulsions. And the nice thing about these nanoemulsions is we can vaporize them in a very controlled manner using ultrasound. So envision you have a liquid droplet that's smaller than 200 nanometers. Okay, it's a liquid droplet. You apply ultrasound, you're changing the pressure locally, and you vaporize. So we're essentially boiling. We're changing the local conditions in terms of thermodynamics of the perfluorocarbon droplet, and ultimately we can boil that droplet instantaneously. Okay? So why would that be of interest? Well, one of the challenges with HIFU ablation is, depending upon where your tumor is located, you want to be able to use the lowest amount of energy, because you can potentially get skin burns. You can get skin burns. You can also get burns at the interface between bone and fat, or bone and muscle. So if you're trying to treat a liver or a pancreas, you need to get past rib cage. The challenge is making sure that you don't end up with burns at those interfaces. So ultimately, we want to reduce the amount of power that's required. Okay? There's evidence that you can reduce the power required as well as the length of exposure if bubbles are present in the tissue. So this was a study that was done by a colleague and published in 2003, where they were able to send a very short high-pressure pulse, get bubble formation in the tissue, and be able to form larger lesions with lower power than if they were to try to do this without bubbles being present. Now, the challenge is, of course, you don't have gas freely circulating inside the body or directly in your tissue. So you need to introduce that either by creating it. In this case, they use a very high-pressure pulse, or you need to be able to inject bubbles that are preformed. And so what we're trying to do is inject, once again, these nano droplets that will be able to escape leaky vasculature, extravasate into the tumors, and then accumulate and be able to vaporize using short acoustic pulses. When I say short, we're talking just about 10 cycles. Okay? Compared to what is typically used for HIFU ablation, which is continuous wave on the order of about 1 or 2 seconds. So very short pulses. So we're able to control the location. So I'll start off by just saying that this is a phantom material that contains these droplets that are dispersed throughout. And you only get vaporization at the focal point. And so by simply moving the focal point, we can actually create an array or a matrix of bubbles inside our phantom material. And the key for the extravasation through the leaky vasculature is to be able to control the size of the particles. So by just using extrusion, we're able to make particles that are smaller than 200 nanometers. And I'll show this video just to give you a sense of how bubbles can actually be used to enhance. This is tissue mimicking material that doesn't have any of these nano droplets. And so the red light is when we're actually sending in or transmitting acoustic energy. So there's a lesion that forms here. This is accelerated by a factor of 10. So it actually took at least about a minute of exposure. This is a small number of particles that are dispersed throughout. You only get bubble formation, once again, at the focus, and your lesion forms in a much faster rate of heat deposition. And you increase the amount of the number of particles, you can also increase the density of vaporized particles and further accelerate this ablation. In addition to that, you can also increase the volume of ablated material. So last thing I want to mention then is we can detect bubbles, or your ultrasound contrast agents are nothing more than coated microbubbles. okay, either coated with a protein or a lipid or a polymer. So we can use BMO ultrasound, or at least that technology, to detect the bubble formation. And the question is that we ask, how well, first and foremost, how well does the bubble cloud and the dimensions of the bubble cloud correlate with the lesion volume? So we're using a imaging probe passively, so we're actually not transmitting BMO ultrasound pulses. We're just listening for energy that's radiated or scattered by the bubbles at the focal spot. Okay, so we end up with, after some signal processing, you end up with an image that looks like this. This is just basically the amount of power that's been radiated over time, and you get a two-dimensional picture of that. And so we can then test the correlation of that from the BMO image with the location of our lesion in real time. So using ultrasound technology, but changing how you use ultrasound technology. So instead of using it as a pulsar receiver, simply using it as a receiver. And it's a 2D array of receiving elements, acquiring the radiated energy that's been generated and produced by bubbles at the focus, and we get a really good correlation between what is actually produced and that you can actually see and measure optically with what's been produced using ultrasound techniques. So you're actually detecting this acoustically. So I'll finish with that. Want to acknowledge, of course, the members of my group. And the last bit of work was actually done by Penzang. And these are my collaborators at Boston University as well as Harvard Medical School. And this is my funding sources. And I thank you for your attention.
Thank you, Professor Porter. Do we have any questions for him? Of course, I should have known it was going to come from the other professor.
I have two questions regarding the thermosensitive liposome. The first one is how do you attach the polylipid PAA copolymer to the liposome surface?
Mm-hmm.
And after you attach it, will that affect the circulation lifetime of the dorsal liposome? The second one is, are these two polymers, the copolymer, are safe for clinical use?
So the first question, we ended up with roughly the same length of time, circulation time, as if we had just used pegylated liposomes. So we actually just we did a biodistribution assay earlier this year for that. The second, the question or the answer to that question is, it's actually a really good question. So I'm not sure if these are biodegradable. So I'm not sure exactly what the response of the body will be over long periods of time. But we did do a toxicity assessment earlier this summer. And for varying the amounts of the polymer, we did not see a change in the body weight for the animal, and we also didn't see any early death from the animal.
There was an earlier report about the polylipid polymer, and this polymer may generate a neuron toxicity.
Okay.
But it's not many reports later.
So that, in terms of toxicity to the neurons, we have not looked at specifically.
I have a question about your emulsion work.
Yes.
One of the effects that I've seen as a potential issue for emulsion therapy with ultrasound in the body is the process of osmobrightening.
Yes.
By small emulsions can
Yes.
dissolve and then add on to the bubbles. I want
Yes.
to know if you had any thoughts on how to deal with that.
So we do pegylate the surface of our emulsions, and that seems to stabilize the size. We still need to do some additional assessment inside, say, serum. And, you know, it would be nice to be able to also do a blood draw and see if there's also any change in the size of the particles. So once you introduce serum, that often changes things in terms of the conditions that you're exposing your emulsions to. But we have not noticed, if we do not pegylate, I will say if we do not pegylate, we do see an increase in the size over time. Yes.
Thank you. Thank you for that interesting presentation. I have a question. If you For the thermal treatment of tumor tissue, if you had a window, an actual window, I think.
Through like the rib cage or?
No, an actual window in the skin.
Okay.
I think that could actually be done for periods of 3, a maximum of 4 weeks without complication for patients. Would that help you in terms of the thermal application?
So are you saying to actually
It'd be a
strip some of the skin?
No, actually put a window in.
Oh,
Just like they, you can put in a percutaneous lead for a long period of time. You can actually make a window. We do it in test animals.
okay.
You could actually probably, I don't think it's been done in humans, but you could certainly develop, you can put almost anything in for up to a month, or 3 months. So I think for a month, you could actually put in something like that. And how, would that help you, or would it not be much of a difference?
I think that the larger challenge is getting past like the rib cage or any other interfaces. In particular, there's been some studies on if you end up with some burns at the interface between fat and muscle. So this is actually inside the body. And there's evidence that you can get burns if the power that's been transmitted is too high. And additionally, trying to get through or past the rib cage. So trying to treat any organs or tumors that are growing in organs that are protected by the rib cage. That's also a challenge. And so our thought is that we can reduce the power that's required for ablation, then your concerns of burns at the interface between the rib and the surrounding tissue is mitigated. So a window from that perspective would not be beneficial.